All right, this is the first PowerPoint video for Unit 2 and our introduction to metabolism. We're going to talk a little bit about energy in this particular PowerPoint, as opposed to jumping right into cellular respiration and photosynthesis. Just to give us a little bit of background, a little bit of understanding why it's important to study those processes and the other ones that we'll be studying. So here's a great picture of energy transference. We've been talking through Unit 1 a little bit about how we have lots and lots of reactions that are happening inside living organisms at any given time, and we understand that all of those reactions are enzyme catalyzed, so this is not a new idea for us. The sum and total of those chemical reactions is what we call metabolism. So we have really had an introduction to metabolism already through our study of enzymes. And as part of that, what we do as organisms is transfer energy. We take in energy as food, various food sources, and we transform that energy into usable energy for us, particularly or ultimately in the form of ATP. I love this little graphic. This is a visual sort of mapped out as a flow chart of all the metabolic reactions that occur in a living system. So it gives us an idea just how complex we are and how interactive those metabolic pathways are. So we can often easily shift from one path to another. We can use parts of a carb to make a particular protein that we need. We can convert carbs into fats for long-term storage. We can do all kinds of conversions for all kinds of different things, to manufacture hormones, to manufacture bone tissue, whatever it is that we need. So we do also understand that there are some things we can't make that we would not have a full metabolic pathway for. And we saw that or visited that last unit when we talked about the fact that there are things like essential amino acids that we must take in. But a lot of the things that our body needs, we can manufacture, and that's what a lot of these pathways are. And a lot of what we take in, we can use to manufacture other things. So a really neat overall visual of some of the, of the majority of the metabolic processes in the cell. All right, so when we're talking about metabolic pathways, and actually we did talk a little bit about this when we talked about multi-enzyme complexes, again, as part of our study of enzymes. Biochemical pathways are the unit of metabolism. So if we're thinking about metabolism as being the sum total of all the reactions in the body, obviously those pathways are biochemical in nature. And we understand that those pathways have multiple steps, hence that idea of multi-enzyme complexes or multiple enzymes where the product of one is going to be the substrate of the other, of the next, in something that looks a bit like this from our graphic. So where the squares are the substrate/products and the enzymes are being represented here in purple rectangles. So we're looking at a series of enzymatically catalyzed reactions going from a starting molecule and ending at a final product where, for instance, B was the product of the reaction that A went through, but is the substrate for enzyme 2 to carry on through that pathway. Again, there's our definition of a multi-enzyme complex. And another graphic for us to look at where we're seeing an enzyme. This time our enzyme is embedded in a bilipid layer. So we're seeing this as part of a membrane of some description, maybe the inside of a mitochondria or maybe some other pathway. or some other membrane rather, and we're seeing that we've got a group of enzymes, so multi-enzyme complex, where substrates are going in, products are being made, they are the substrate for the next enzyme. So just another visual for that. Which brings us to the idea of bioenergetics. How do we manage our energy resources? And hopefully we've kind of already touched on the idea that it's via our metabolic pathways. So how we manipulate or maneuver the raw material that we have to be able to do the things we need to do. We've got different kinds of reactions, and this is not new. These words coming up are not new for us either. We have catabolic pathways, meaning that we have pathways that can break things from more complex to more simple, and we have anabolic pathways where energy is used to build more complex molecules from simpler ones. Again, we saw this idea in Unit 1 when we were talking about creating polymers from our monomer units in our biomolecules. And the diagram here is a mitochondria, and we're seeing a partial reaction here, a pyruvate oxidation into a lactate molecule, which would be lactic acid, what we would know as lactic acid. So basically what would happen if we didn't have enough oxygen there to complete the pathway of cellular respiration. So we're making lactic acid. And again, that would be just an example of one of the metabolic pathways that are carried out in our body. So again, I think this is a good recap of what you saw in Unit 1 with the amino acids making proteins, carbs making things like, well, amongst other things, glycogen, fatty acids being used to make triglycerides. That is an anabolic reaction, although it does not form a polymer. Well, remember that's a composite molecule, the triglyceride. And carbs, and this is talking about transforming carbohydrates into fats. So be careful here. We don't take glucose molecules and make them into, we're not talking about the above pathway, basically, where we're taking carbohydrates and making them into a longer term storage. We're actually transforming them. There's too many for glycogen storage, and we're transforming those into triglycerides. And we will be looking at that pathway later on, how if we take in too many carbs, we convert them to fat and store them that way. And we'll look at that process. So those would be examples of anabolic reactions. And we know if we reverse those reactions and broke those things into their smaller units, they would be catabolic. We've also looked at this idea of energy, and I know you guys do a little bit of studying of this in physics, and maybe you were or could sort of superimpose that on the idea of enzymes and what they're able to do for us. But just as a recap or as an intro for people who haven't done physics, kinetic energy, in this diagram we're showing energy being put in, so energy in motion or energy in change, potential energy at the top of the hill, and then energy out, which is also kinetic energy. So let's look at what energy is in general: the capacity to do work or ability to cause change, and that's a pretty standard definition no matter what science subject you're studying in. And we also refer to the two kinds of energy: potential energy and kinetic energy, and they apply as much in biology as they do in physics or chemistry. So potential energy is stored energy. The energy is there for us to use, but the change, any change is not occurring at that time. So think of maybe stored carbs stored as glycogen in our muscles. It's stored there, ready to be converted back into glucose and then used to make ATP, but not being used at the present moment. Where kinetic energy would be energy that's causing change. So again, we may be converting glucose into ATP so that we can do what this guy is doing in the picture and pedal our bicycle. All right, back to the idea of potential energy again, stored energy. And in living systems, that energy is usually stored as a kind of bond energy, which is the energy, for our purposes anyway, and this may not be exactly what you did in chem, but for our purposes, we're going to interpret bond energy as energy required to break or form a chemical bond. And that energy can be easily converted. This is a cool little picture, again, illustrating that idea of potential and kinetic energy. We're seeing that the guy climbing up the stairs is using his muscles to get up there, and so he is demonstrating kinetic energy, or we're seeing kinetic energy there being used. Up on the platform, we've got a higher potential energy. The diving process or jumping off process, he doesn't look like such a great dive yet, is going to be an example of kinetic energy. And once he hits the water, he has a much lower potential energy than he did at the top of the platform here. So when we're talking about those energy transformations, we're really talking about the study of thermodynamics. And again, we won't be going too far into this idea, but we will touch on a few of the principles here. So you're probably aware there are two fundamental laws that govern the idea of thermodynamics, and they are called the first and second law of thermodynamics. The first law is about energy, the fact that energy cannot be created or destroyed. Again, that for some reason that seems to be one of those things you hear quite a lot, sort of through just life in general. Here's a nice biological example of that, probably not one you'd see in physics. The chemical potential energy in the food is going to be converted into energy that's going to be used to move this cheetah, right? Cheetah, the fastest land animals. So lots of kinetic energy required for them to get around. So the energy here is being transferred and transformed. It's being taken in by the cheetah, and then it's going to be converted into energy that's required for the second law of thermodynamics is all about chaos, which is kind of a neat idea. And the idea that disorder is kind of the state to which we are always moving. So the idea that disorder, or what we call entropy, is continually increasing. And I like this little graphic at the bottom of the bedroom. I don't know about you guys, but that tends to happen in my house. Disorder tends to, or seems to, happen spontaneously, and then it requires lots of energy to reorganize that particular space. So energy does transformations do proceed from a more ordered, less stable form to a less ordered, more stable form. So it's more stable to have chaos and less stable to have this sort of organization or order. So how does this apply to biological systems, and why is this an important? It may seem that we sort of go against that rule, if you like. And we're going to look at some points that relate to why it is that we can say that we're not sort of breaking the second law of thermodynamics. So a few points. We understand that that in energy conversions, lots of heat is released into the environment. This actually makes the environment more disordered. So some of that extra energy that we give off as waste product is going to cause more disorder in our environment. We are always converting energy into heat, and every energy change or transformation that increases entropy or disorder of the universe, or each one of these kinds of changes is going to do that. By contributing the heat to the environment, we're increasing the entropy of the environment around us. And this graphic does a much better job than my rambling here. So we've got our cheetah again, and as the cheetah is racing off perhaps after that antelope or deer that he was eating, the cheetah is giving off heat into the environment, also giving off small low energy particles or low energy compounds like carbon dioxide and water into the environment. So even though the cheetah stays nicely ordered, the environment around him is suffering as a result of that. So he stays ordered, but the environment is brought closer or farther and farther to the side of disorder or entropy. So to sum that up a little bit, cells create order. We use energy. We transform our food sources into particular molecules that will allow us to become more ordered. However, we do not violate that second law of thermodynamics. In fact, we increase the disorder, increase the entropy in our universe. So if we look at the system as a total, us with the rest of the universe, we are actually as a unit moving toward disorder. So we are not defying that law. So by ejecting, I guess, for lack of a better word, our waste products into the environment, we are increasing entropy in the environment, while we are decreasing entropy within our own system, at least until we die. And then, of course, our system becomes, or what was left of ourselves as a biological system will become more disordered. So, as we said, we increase the entropy of the universe by maintaining our own structure. So, in order to describe this, or one of the ideas that are used to describe this is free energy. And we're going to have a little bit of a look at what free energy is. Here it's defined as energy that can do work under cellular conditions. And we live at the expense of free energy. So it takes a lot of energy to maintain that cellular structure. So free energy was a concept that was explored by a guy called Gibbs, and you may have heard of Gibbs free energy, probably worked with it in chemistry if you took chemistry. So free energy is, for us, the energy that's available to be able to do cellular work, which really involves making and breaking chemical bonds, so forming and reforming materials in the cell that we require. And as I mentioned, it's expressed as Gibbs free energy. There's a little bit about it in the textbook. There's a few examples in the book about Gibbs free energy that are not too bad. I won't be asking you to do much with Gibbs free energy, but just to understand the idea that the energy is available to do work for the cell/organism involved. So in a cell, the Gibbs free energy is going to refer to the amount of energy that is contained in the chemical bonds there, assuming constant pressure and temperature. Any change in free energy is expressed as delta G. And any reaction and we've used these terms before as well, any reaction that requires an input of energy is considered to be endergonic, meaning that we need to add some energy, free energy, to that system, and exergonic, meaning that energy is released. And usually in living systems, what happens is we couple these kinds of reactions. So if we have a reaction that's going to require an input of energy, we also carry out a reaction that is going to release some energy. So we do a lot of energy transforming in our cells that way. We couple endergonic and exergonic reactions to be able to manufacture the materials that we need to be able to maintain order or make sure that the cell does not move toward greater entropy. So a relook at an exergonic reaction. We understand in exergonic we're able to release energy. So if we're looking at free energy totals, and this graph is showing free energy along the along the y-axis there, and progressive reaction along the x-axis. So going from the reactant on the left with lots of energy to the product on the right, and we can see that energy is being released in that reaction. So this is what we would call an exergonic reaction. Energy is released. The reactants have more energy than the products. So we're releasing energy usually into the atmosphere, into the ecosystem. These tend to occur spontaneously without adding energy to the system. Please note, though, in a biological system, these are still enzymatically catalyzed reactions. We need to make sure we keep the heat down in living systems. Okay, we're going to have a look at a similar diagram that is involving endergonic reactions rather than exergonic. And in this same kind of graph, we're seeing free energy on the y-axis and progressive reaction on the x. However, in this case, we see that the reactant has a lot less energy than the product, and therefore energy must be added to the system. That's what an endergonic reaction is. The reactants have less free energy than the products, so energy must be available to put into this system. So we're creating organization here and decreasing entropy. Again, these do not occur spontaneously. These ones do not occur spontaneously because we need to add energy, but just as we saw in our exergonic reactions, we also require an enzyme for this to occur. And again, I mentioned earlier, often we'll see that endergonic and exergonic reactions are coupled so that the energy from one reaction is able to be used to help the other reaction progress, or to make the other reaction, or give the other reaction the energy it needs to happen. Okay, so here's a little graphic that sort of compares the two, endergonic and exergonic. Take a minute to have a look at what the diagrams are sort of illustrating to you. Same thing that we saw before. In the endergonic side, we're seeing a reactant that has low energy compared to the product, so energy must be supplied. And on the exergonic side, we're seeing a reactant that has higher energy than the product, so energy is released. All right, we have talked about the idea of equilibrium before, sort of equal forward and back rates for a buffering system. So we're using the term equilibrium here in a little bit of a different way. The reactions, the sum and total, not a single reaction or not a single movement, but the sum total of reactions that are occurring in a cell in a living system are not going to be able to, or not ever going to reach equilibrium in all, sort of with all of the reactions, if you're looking at it that way. They're what we call an open system. So we are in constant need of supplies from the outside world. If we're thinking about it from the perspective of the cell, we're going to be using things that are brought in, and therefore we're always going to find that there's a need, a supply-demand idea going on here. And as we're metabolizing, using up the things that are being brought into the cell, we produce waste that must be removed from the cell. So this is an open system, and we always have things flowing into and out of the cell. And this must be the case in order to maintain that order that we were talking about earlier in the cell. So because this is the case, we are never really going to reach metabolic equilibrium in terms of cellular reaction. Okay, this is a little graphic that's used to describe in general the process of cellular respiration, which will be our next big topic area, where we break glucose down to form ATP. And it's showing basically that the material is always flowing in and material is always flowing out. So we have a constant movement of material. In this case, we're going to be taking glucose and putting it through significant changes to yield ATP. And that is an ongoing process. As long as we are alive, we require the formation of ATP to be able to do our cellular work. All right, so we're going to revisit this idea of energy coupling. As I mentioned before, coupling exergonic and endergonic reactions are how we get things done in our cells. And in the graphic on the side here, we're seeing on the left-hand side, follow the light green to the darker green. This is meant to be the process of cellular respiration. So the process of cellular respiration yields energy. So it is an exergonic reaction. It's actually an exergonic series of reactions, many, many reactions within that umbrella of cellular respiration. So glucose metabolism, they're saying here, breakdown of glucose, which is cellular respiration, is an exergonic reaction and energy is being released. The energy that's being released from those reactions are being used in a endergonic reaction, which is the synthesis or the condensation reaction that occurs when we form ATP. So if you look at the ATP in the center, you can see that from the bottom we have ADP, so adenosine and 2 phosphate groups here, ADP, and there's a phosphate just kind of hanging out there. We usually call them PI, inorganic phosphate, because they're no longer connected to a carbon backbone. And we're going to use the energy that's released from the breakdown of glucose, so again, coupling that left-hand side reaction with creating ATP, so it's a condensation reaction, again, all enzymatically catalyzed. We're using the energy from glucose, exergonic reaction, to create ATP from ADP, which is an endergonic reaction. Then we're taking that high energy molecule and having a look over at the right hand side here, and we see that we're going to do the same thing. We're going to carry out the hydrolysis, the breakdown of ATP, and in doing that, that's an exergonic reaction. We're going to use that energy to take amino acids and form them into polypeptide chain, which is an endergonic process. So we've got a double coupling here. On the left, we're building ATP, which is endergonic from the exergonic glucose metabolism. And on the right-hand side, we're building amino acids into proteins, an endergonic reaction, an energy-requiring reaction by breaking down the ATP, which is an exergonic reaction. So 2 examples of coupling here. So we've looked at ATP before. We understand that it's our major energy molecule in living systems, so in the cell. This is the structure. We've looked at it before. It's a nucleotide. If you remember, it has the ribose sugar and the adenine nitrogenous base. And then instead of just having 1 phosphate group, which we expect in most of our nucleotides, we have 3 phosphate groups. And clear to see in this picture, we've got lots of negatives here. So the bonds that occur between these phosphate groups are actually high energy bonds. It takes a ton of energy to push all those negatives together, which is great for us because that means that we can store a lot of energy in those bonds, and then we can release that energy when it's required for other reactions to occur. So this graphic is showing us the hydrolysis reaction, as I mentioned earlier, of ATP into ADP. And water in the presence of an enzyme is added in, breaks the bond between the 3rd and 2nd phosphate group, and energy is released. And again, we'll usually couple that energy or use that energy to carry on an endergonic reaction, one that requires energy to be added, and ATP will be reformed again by glucose metabolism generally. All right, we've been talking about cellular work. What kinds of things do we actually mean by that? So in general, we can divide the kinds of cellular work into 3 different large categories: mechanical work, transport work, and chemical work. And there's a few graphics that we're going to have coming up that will give you maybe some examples of that. So transport we looked at already, of course. This example at the top of the graphic here is showing materials being transported from an area of low concentration to an area of high concentration, so against the concentration gradients. And we can clearly see here that the phosphate molecule has been attached to that membrane protein, allowing energy for that reaction to occur. So for those molecules to be able to go against the concentration gradient from one side of that membrane to the other. There's a middle example here of mechanical work, and they're talking about energy being used here to transport particular molecules. They're calling them motor proteins. being able to move proteins around in the cell. And the bottom one is supposed to be an example of chemical work where we're using the phosphate group as an energy source to couple whatever this X and Y might be. So again, maybe one amino acid to another amino acid to form a dipeptide, and phosphate is being used there along with an enzyme to make that happen. So energy coupling, a big idea here. And there's another example of sort of the same thing. Maybe I've beaten you over the head with this. So lots of different graphics showing you exactly the same thing. So that idea of the ATP cycle, ATP to ADP to ATP to ADP, keeping in mind that we need energy to reform ATP from ADP, and that we are able to release energy to use for work when that last phosphate group is removed in that hydrolysis reaction. I mentioned this already as well, and we saw it in our diagrams that we just looked at. ATP drives those energy requiring reactions by phosphorylating or transferring a phosphate group onto some aspect of the molecule that's involved in the reaction. So when that happens, we say that the molecule has become phosphorylated, and we understand that that means that we're adding energy. It's like plugging in a power pack. So the graphic on the side is showing the transformation of certain amino acids into other amino acids. And we, again, understand that other than those 8 essential, we're able to transform within those amino acids to make the ones that we need. So in this case, we're seeing that we've got an amino acid. Say, let's take the top example, serine. And ATP is being used to phosphorylate the serine in what would be an intermediate step leading to something else. So this is only one step in the process, but it's our energy requiring step. And in each case, we're seeing an amino acid being phosphorylated in preparation for whatever the next step in this pathway is. All right, and here's that similar diagram again, same kind of idea. And again, in each case, if you look at the diagram, you can see that molecule has been phosphorylated, and that's how we're transferring that energy through the hydrolysis of the last phosphate group off the ATP. All right, that is the end of our PowerPoint. A lot of slides and a lot of words, but only a few ideas there. Have a look back over it and see if you have any questions. Please bring them to class on the assigned day.